Good afternoon, everyone, and welcome to today's Epicenter Psychosis Speaker Series. I'm delighted to introduce today's distinguished speaker, Dr. Bill Hetrick. Dr. Hetrick is a professor and chair of the Department of Psychological and Brain Sciences at the University of Indiana, as well as one of the co-directors for the Clinical and Cognitive Neuroscience Center, also at Indiana University. His work focuses on understanding risk factors and mechanisms underlying schizophrenia and other related serious mental illnesses here, and has relied heavily on different psychophysiological assessments as well as imaging techniques to try and decipher and clarify the processes that are unfolding in these illnesses today. Maybe even more importantly, at least given the setting of today's talk here, Dr. Hetrick is one of our most distinguished alumni of the clinical psychology graduate program at The Ohio State University, which makes it especially exciting to have him joining us here today. As always, questions for Dr. Hetrick at the end of his presentation can be entered in the chat, sent to me, please. And we will have some time at the end for a Q&A session with Dr. Hetrick. Closed captioning is available by clicking the CC button on the bottom of your screen. And with that, I'm delighted to turn things over to our distinguished speaker, Dr. Bill Hetrick, to hear about the really amazing work that his lab has been doing, trying to demystify the role of the cerebellum in schizophrenia and highlight the importance of this long overlooked region of the brain. Thank you, Dr. Hetrick.
Dr. Breitbart, thank you very much. Appreciate that very warm and extremely generous introduction, Nick. Let me get my slides up and see, get going. Okay, I do that. Are you seeing the slide presentation as a single image or multiple slides?
Multiple. It's as if the slideshow is not started. Just the PowerPoint is open.
How's that?
That's perfect.
Okay, thank you. So I've heard from my graduate students about this terrific group that you have meeting, and I'm embarrassed that this is the first time I've come. And so I will make an effort to join. I really enjoy hearing from the students about what's happening in this group and the exciting work that's happening in the field in schizophrenia. So thank you for organizing this, and I appreciate the work you guys are doing. I do want to talk, as Nick said, about the cerebellum, and I want to make the argument that it's not really the lesser brain. It's an important part of the brain. It's an important node in a distributed network, and I think understanding that node is important. I don't want to elevate this node above all other nodes. I just want it to be included in discussions and vetted as a region that may be implicated in psychotic disorders, and schizophrenia in particular. So I want to focus on the function and some structural demonstrations of the cerebellum. In terms of the history, as Nick said, I got my PhD at Ohio State, and I wish I had driven underneath this welcome to Ohio sign this year to be in person at Ohio State. But of course, there's some benefits of this virtual world we're in now, and one of them is that we can have such a varied group in attendance today. But I did find my PhD at Ohio State, and I appreciate that. I wanted to point out that in '99, when I got my degree, I got it from David Smith, who had received his PhD at Stony Brook from John Neal. John Neal had received his PhD at Vanderbilt from Rue Cromwell, And Rue Cromwell was another distinguished PhD alum, and he had received his PhD from Julian Rotter. And Rotter had been a student at Indiana and received his PhD under Chauncey Lowett, who received his PhD from Robert Yerkes, who received his from Hugo Munsterberger, who received his from Wilhelm Wundt. So this whole exercise of understanding academic genealogy happened when I was a graduate student at Ohio State. Professor Hall required us to do this in the history and systems class in my graduate program. And I've taken an interest in academic genealogies, and by showing mine, I'm hoping that it'll interest you in yours, and you can learn a little bit more about your academic genealogy, the interesting pieces of history that can be found in these genealogies. I want to also begin by thanking my collaborators and students. These include faculty, in particular Brian O'Donnell, and Olaf Sporns, Charlene Newman, Ina Hughes. Mandy Bolbecker has really been instrumental in leading our lab through a variety of grants that we've worked on. Graduate students, this is an older photo of one of our infamous road trips to a conference, and then other colleagues and former graduate students as well have played important roles. And you'll be hearing their names as I present some of the data today. I want to focus really on three things. If you have to leave early, or if we don't get through the entire slide presentation, I'll summarize the important points as we go through these three questions. So what, in my view, and there are different theoretical models, conceptual models you can bring to bear on research questions and psychiatric disorders, or in a more Ardochian fashion, psychological constructs. And the view that we have brought to bear on this is to think about what are the types of fundamental disturbances that give rise to psychotic disorders, or schizophrenia spectrum disorders in particular. And in our thinking, the discoordination of cognitive and motor processes is a real important part of that fundamental disturbance. And I'll tell you why I think that this fundamental disturbance may be associated with a discoordination problem, and what role the cerebellum plays in that coordinative or discoordination problem in schizophrenia. The second thing I want to sort of answer is a question that says, well, then what brain systems may be involved in this fundamental disturbance? If you take this approach that discoordination of mental processes is fundamental to the disorder, what are the brain systems involved? And the answer, as you know, is going to be cerebellum and its connections and interactions with the cerebrum. And then finally, I want to talk a little bit about, can we target cerebellar mechanisms to improve associative learning, a cerebellar form of learning that we'll discuss in this presentation. And the answer is maybe. There are two promising psychopharmacological trials that we've engaged in that suggest that maybe some of the mechanisms that we're speculating are in fact involved in the disorder. So those are the three directions that we want to go, and the talk will be organized to answer these three questions. Now, in terms of the first question of this fundamental disturbance, I'm taken by Bloiler's characterization of the disorder. Kraepelin described what he saw, dementia praecox, early cognitive decline. And Bloiler described what he thought was happening, a theory about the disorder. And so he coined the term in 1908, schizophrenia. Greek roots are schism, to split, and frame the mind. And he postulated a breaking of the associative threads between ideas. He postulated really that the core deficit was a fundamental associative disturbance, fragmentation of cognitive processes, mental processes. And the loose associations, the derailment, the loss of goal attainment, the tangentiality, the perseveration, blocking, and so on, were byproducts of this fundamental associative problem, indications of it. And certainly hallucinations and delusions, sort of higher order, more abstract problems, were byproducts of this fragmentation of the cognitive process. And so this is sort of the fundamental nature of the disturbance that we see, and at least the lens that we're taking. As I said, there are a number of theoretical and conceptual frameworks that you could take. We happen to be taking this one, and for this reason. A contemporary of Bloyler's, Erwin Stransky, came up with this idea that maybe schizophrenia and dementia praecox, this syndrome of disorders, could be characterized by intrapsychic ataxia. And that's really interesting. He draws this analogy from the motor system and says that what he was observing was this notion that this discoordination in this the intrapsychic domain was analogous to the discoordination in the motor domain. So motor ataxia is the coordination disorder that affects the direction and amplitude of voluntary movements and perturbs voluntary muscular contractions needed for postural adjustments and maintenance of same positions. And he thought that intrapsychic ataxia was being observed, and it was being observed as inappropriate, sort of goal inappropriate, inverted, irrational, and interrupted at times by perseverance and inappropriate actions. And this does map on conceptually fairly well to what people think the cerebellum does strictly in the motor domain. Nancy Andreasen, I'll come back to that actually, Nancy Andreasen began to really propagate this idea that is these strong historical roots with her concept of cognitive dysmetria, which I think most of you would be familiar with. Again, another analog of the motor system, difficulty in coordinating the processing, prioritization, retrieval, and expression of information. Interestingly, Kendler did this review where he looked at, he identified and reviewed clinical descriptions of schizophrenia or dementia praecox appearing between 1899 and 1956. 18 types of diagnostic, categorical types of systems, and compared them to the 6 modern operationalizations. And the findings were that odd movements and postures were noted in 16 of the 18 historical pieces. Those pieces are important. They ended in about 56 before neuroleptics became prominent. So these motor problems, odd movements, were present in the absence of neuroleptics, which we know have profound, at least the traditional neuroleptics have profound effects on motor processes. And he wrote this interesting conclusion. While abnormal motoric functioning is well known to characterize catatonic schizophrenia, nearly every one of the authors describes subtler motoric abnormalities, often posture, gesture, gait, frequently seen in schizophrenia. And because nearly all of these authors were writing before the widespread use of antipsychotics, these symptoms can't be drug-related dyskinesias. So if I'm going to talk about the cerebellum and make a case that the cerebellum is involved in schizophrenia, you probably would expect that part of that argument would say, yeah, there are motor problems in the disorder, in the psychotic spectrum. And the answer is yes. Vijay Mittal is doing this. It follows a rich line of history that Elaine Walker began. A number of her students are pursuing this. To sort of drive home that point, the motor system, the motor abnormalities, you see a delay in milestones achieved. You see a delay and worsening performance associated with subsequent measurements at later ages in very large cohorts. And you see these disconnect-like problems on spontaneous movements that Elaine and Vijay have studied a bit more extensively in these medication-naive first-degree relatives or first-episode relatives in schizotypal personality disorder. So that the motor system is impaired. And this slide illustrates the effect size. So this is the Cohen's D effect size. And if you just look up here, you see which ones are the strongest. This neuromotor deviation shows a very strong effect size. In this particular study, the strongest effect size. And we'll see in a couple of other instances in the presentation that the cerebellar-based effects have fairly large effect sizes compared to traditional things. So here's ventricular volume, one of the first and earliest indications of morphometric problems in schizophrenia. And these neuromotor deviations are very prominent, even compared to what we think of as maybe some of the most profound structural problems in the disorder. This issue of the cerebellum and the problems that have been observed with the cerebellum is nicely illustrated in this 2017-2018 paper by Mulberget. It has 983 patients and 1,300 controls, and they examined this in a meta-analysis of 14 fairly large studies. And what they showed was that there's a cerebellar cerebral structural covariance that can be demonstrated. And they began by showing that there is reduced gray matter in schizophrenia in cerebellum reduced gray matter. And I don't want to dwell on this slide, but these are the effect sizes of that gray matter reduction across the 14 studies, showing a pooled effect size here. And then this is indicating the gray matter volume reduction in schizophrenia compared to controls. It's always humbling to see these large effects. These are fairly large effects compared to other effects reported in the disorder, and recognize how much distributional overlap there is. But this figure illustrates that. Interestingly, this cerebellar matter reduction, gray matter reduction in schizophrenia, was most prominent in regions that show functional connectivity with association regions in cerebral cortex. So you can take the cerebellum and ask, what part of this, where is this part of the cerebellum connected with the cerebrum? And these maps, this particular map was developed on a sample of 1,000 participants, as the paper describes. And what they show is that those regions of cerebellum where there's gray matter reduction are connected with frontal regions where there are also cortical thickness problems and reductions in gray matter. And in particular, they show these in the order of the connectivity. And so the frontal regions of connectivity with the cerebellum's connectivity with frontal regions, I should say, is where the effect size is the largest with regards to the covariance of the gray matter reduction. And this indicates that the problem with cerebellum is probably not just cerebellum, it's the connectivity with cerebrum. And you could say that in a reverse order. You could say the problem that we see in cerebrum is probably not just a problem with cerebrum, it's the closed loop circuits that exist between cerebellum and cerebrum, where the cerebellum is playing this role of guiding the neural processes using predictive models that I'll describe here in a second. And we'll see more of this, that this connectivity between cerebrum and cerebellum seems to be reflecting that the problem is in the circuit, not that the cerebellum necessarily is driving the problem in the cerebrum, or not that the problem in cerebrum is driving the evidence that we see in cerebellum, but rather perhaps some problem in the circuit is affecting both cerebellum and cerebrum. Finally, the effect size of these cerebellar problems, the cerebellar effect sizes are comparable in magnitude to the most consistently reported problems in cerebral cortical regions and subcortical regions of the cerebrum. And you can't read this, I don't expect you to read it, but the effect sizes are here, and you can see that what's being shown are those regions that have stronger effect sizes. And I've simply highlighted the regions of the cerebellum. This happens to be that ROI6, which is the frontal parietal regions of cortex that are connected with cerebellum. And this is hippocampal volume, if you can read it at the top. And I just wanted to show that these cerebellar regions are scattered amongst regions such as hippocampus and insula and amygdala and so on, regions that have been identified as being problematic. What you can take away from this is that the cerebellum isn't some odd structure that has modest differences compared to these other prominent structures that are studied quite heavily in the field. Instead, the cerebellum is performing, particularly these regions that are connected with frontal regions of the brain in closed loop circuits, seem to have robust effect sizes. So I've shown you evidence of motor problems, and I've said that these motor problems can be seen very early in life. They can be seen in naive, medication naive, early psychotic episodes, and so on, in family members, in fact. And I often think, the lab thinks about this in the context of the neurodevelopmental origins, that if you look from conception through birth, infancy, and so on to adulthood, we know that there are genes and pre and perinatal complications that affect these neural systems, frontal systems, as I've been discussing, dopamine systems, stress responsivity systems, and so on. And these prominent models, this one from Tykanen, shows that these motor abnormalities seem to be seen fairly early in this trajectory. And what's important about that is that there's evidence from developmental psychology, particularly from folks like Linda Smith and Esther Thieland, who's passed now, but here at Indiana, that these developmental theories argue that there is no distinction between motor and cognitive processes. People who study kids and study development, when they look at kids, when they study kids, they say that the motor system and the cognitive system are indistinguishable. They don't understand people like me or us that say, well, there's motor processes and cognitive processes. And they argue that these very early motor processes are providing the scaffolding by which the nervous system can build more complex processes using the same fundamental architecture, essentially, the same fundamental mechanisms of building out that architecture, to then expand on that architecture for cognitive processes. And they take somewhat of an embodied cognition view, which sees cognition and an embodiment or the manifestation of those cognitive processes within motoric and bodily functions. They take that type of view of it. This was something that's been somewhat influential in my thinking about the relationship between motor deficits early and cognitive deficits later. And there's an interesting paper by Riddler on this that shows that early life motor problems in a prospective study where they're examining individuals at 7, 8, 9 years of age, that those milestones that they measure in motor systems at that young age predict cortical thinning in adulthood and predict in very modest ways, such as Elaine Walker's modest correlations between childhood motor problems and the development of schizophrenia, that these motor problems predict schizophrenia onset and cortical thinning in a variety of regions, including these cognitive regions. So this is sort of the framework. For that first question, what's the fundamental disturbance? It's this coordinative problem that can be manifest in the motor system, and that I've not shown you a lot of evidence of motor dysfunction, but of cognitive dysfunction, I should say, but that's the disorder itself. It's the tangentiality, it's the thought disorder, and so on. So what brain systems may be involved? Well, I've already begun to talk about the cerebellum quite extensively, but is this really the lesser brain, I should say here? I don't think so, because the cerebellum contains at least 50%, some estimates put it at 80% of the brain's neurons. So it's 50 to 80% of the brain's neurons in the cerebellum. They're very small, they're very fast, they're complexly organized, and they're able to do extraordinary computational processes. The connectivity between the cerebellum and the cerebrum is extensive in these fiber tracts, just extraordinary fiber tracts, which are in fact very difficult to disentangle using tractography. But they connect the cerebellum in typically closed loop circuits. That a region of the cerebellum is typically connected with a different region of cerebrum in a closed loop, often going through thalamus and so on. But each area of the cerebellum is probably providing the same computational capacity to a variety of regions in cerebrum. 50% of cerebellar functionality is coupled with cerebral association areas, including cognitive control and default mode networks. And this is the work of Buckner. It shows that the percentage of the cerebellum, the brain networks associated with the largest percentage of cerebrum have the greatest cerebellar representations in this homotopic relationship. And this is sort of illustrated in this positive correlation here, showing the percentage of cerebrum and the percentage of cerebellum. And again, this is similar to the Moberget paper that I just showed you structurally, showing that there's this structural covariance between cerebrum and cerebellum. And there's also this functional covariance or homotopic relationship between cerebellum and cerebrum. So again, I want to emphasize for those of you who don't study cerebellum, but maybe study cerebrum, that there are probably regions of cerebellum that your favorite region in the cerebrum is connected to. And the cerebellum is providing important computational contributions to that region for you. Now these corticocerebellar circuits have been described by a number of individuals. If this stuff interests you, Jeremy Schmaumann in the Harvard system has studied this extensively in stroke patients, where he's just been interested in strokes, and particularly cerebellar strokes. And he finds that people who have cerebellar strokes develop something called a cerebellar cognitive affective syndrome. If you were to just go read that paper and look it up, it is a textbook definition of the neurocognitive deficits you see in schizophrenia. Nancy Andreasen has pushed this, as I've said, this corticocerebellum circuit. And then Peter Strick at Pittsburgh is a major player in this area. And he's pointed out that the cerebellum plays a fundamental role in timing. And so we study temporal processing deficits and have a number of papers showing that time estimation, time production deficits in schizophrenia. Sensory motor imagery and representation and integration is an important thing for the cerebellum does. And then this machine learning and predictive coding part of the cerebellum that Vijay Mittal's group at Northwestern has been integrating into their work on early prodromal phases of the disorder. Let me just illustrate through this paper by Ramnani what we think the cerebellum may be doing for cognitive areas of cerebrum. But let me start by showing sort of the textbook example that you would see in a neurology course in medical school about what the cerebellum does. And let's start with a theoretical model of motor function. So this particular model just says that there's a motor command that is sent to the motor system. When that motor command is sent to the motor system uses the musculoskeletal system to create a motor output that has sensory consequences. Turns out that that motor command provides an efference copy. And that efference copy goes into a forward dynamic model that creates a prediction about what's going to happen. The predicted sensory consequences then can be compared to the actual consequences. And if the reafference and corollary discharge coming from these signals conflict, then there's an error signal that goes up and updates the system. And there are connections between these such that, you know, if I close my eyes and go to my nose, or in the inebriation test that you see along the roadside, when people are doing this, there are adjustments that are being made. Or if you're doing the Romberg task and putting one foot in front of the other, there are predictions being made and adjustments being made by this system, theoretical system, to coordinate these motor processes so that you can swing a baseball bat or a tennis racket so that you can ride a bicycle, so that you can engage in these motor activities like reaching for, a glass of water and so on. Now, how is that instantiated anatomically? So this is what happens anatomically. The premotor cortex is sending its signals to the spinal cord and the musculoskeletal system. It then generates motor output and sensory consequences. Turns out that this efference copy is going to the cerebellum, and in particular, the cerebellar cortex is creating very intricate computations and models, and then the output of those go through the deep nuclei and create a predicted sensory consequence and motor consequence. These are compared probably at the level of the spinal olivocerebellar pathways. And if there's an error signal, there's an updating of the cerebellar models. And these models then exist sort of in perpetuity and allow us to just jump on a bike and be coordinated, or allow us to pick up a racket and hit something if we practice that. But if we have a heavier racket, or if the circumstances change, we need to update those systems. And that's what this particular model does. So finally, what might be happening then at a cognitive level? The theory is that there is some, the cognitive regions, the prefrontal cortex regions, there's some information arising from the prefrontal cortex is copied to the cerebellum in the same way that the motor commands are copied from the primary motor cortex to the spinal cord. In this scheme, Romnani argues, cerebellar forward models mimic the input-output relationships of prefrontal targets, or targets in other regions of the brain. This is just an example of the prefrontal targets. Forward models might therefore be able to mimic information processing that's intrinsic to the prefrontal cortex. If you extrapolate from this, delusions may be caused by impairments in forward model systems that predict the consequences of one's own actions, whether they're motor or non-motor in nature. And it could be that hallucinations may be associated with incorrect modeling of external exteroceptive processes that are trying to be integrated in the nervous system. And this is the model of how that might happen. So this model basically is arguing that the cerebellum is really important in creating forward models that guide, coordinate, and allow us to have this goal-directed, smooth, fluid, cognitive process, and not have intrapsychic ataxia. And again, people like Vijay Mittal and Jessica Bernard have used this model in their thinking as well about cognitive functioning in the prodromal phases in high-risk populations. So within this conceptual framework, within this anatomical framework, within this functional framework of the connectivity between cerebellum and cortex, cerebrum, we're using a number of tasks that are cerebellar in nature to find out whether we can begin to understand specific networks. It's one thing to show that behaviorally they're impaired on a task that we think is cerebellar. It's another thing to show that there are gray matter deficits in cerebellum, maybe functional activation deficits in cerebellum with regards to resting state networks. But it's another to begin to map on behavioral problems with the neural substrates that in fact lead to those behaviors. And that's what we're trying to do with some simple tasks. So Eiblin conditioning is one of them. If you told me when I was in graduate studying Eiblin conditioning, which I thought of it even in graduate school in 1999 and so on, I would have thought that I'd lost my mind somehow or another. But it turns out that this is a very powerful technique because it's conserved across species. So Pavlov's dogs, rats, rabbits, humans, other primates, there is tremendous conservation of this response. And what it is that we present a tone, 400 milliseconds of duration, beep, and then we co-terminate with an air puff. So beep, poof, and a puff of air to the eye. And the air is presented, we're measuring the neuromuscular response. Tones are going into the ears, and our signal is an EMG signal in these early studies I'll be showing you. We present a number of these trials where we pair these two stimuli, and in fact, over time, the individual begins to blink in this period before the air puff comes on. As I said, it's highly conserved across mammalian species, and it's excellent translational procedure. The neural circuit is also fairly well described, and as far as we can tell, based on lesion studies in humans, for example, that come from strokes, the network is very well conserved as well. Briefly, the tone comes through pontine nuclei, up through climbing fibers, mossy fibers, and parallel fibers. And at this cortical region of the cerebellum, this is the cortex, this is the deep nuclei of the cerebellum, you get a co-occurrence because the air puff is also being delivered, climbing up through the climbing fibers, intersecting with Purkinje cells. And there's a long-term depression phenomenon that happens here, and then there's a long-term potentiation phenomenon that happens here. And in particular, the Purkinje cells are chronically or tonically inhibiting the deep nuclei. So the deep nuclei don't want to be activated. So if an air puff comes in and a tone comes in, the deep nuclei can code the co-occurrence, but the Purkinje cells need to release their inhibition on the deep nuclei in order for the deep nuclei to generate a blink response that avoids the air puff once the system knows about the timing that the cortex is providing. So we think that the initiation of the response occurs as a result of long-term potentiation associated with the deep nuclei. But the gain function, the amplitude of that response, and in particular the timing of that response, is as a result of long-term depression that's occurring in these Purkinje cells, inhibiting the Purkinje cells so that they release their inhibition so that the deep nuclei can do their thing and generate the blink. The circuit is more complex than that, but that's a beautiful thing if you're trying to understand the circuit. And I'm not going to dwell on this, but it's more complex. There have been evidence of eye blink conditioning deficits in schizophrenia. This technique, as I told you, sort of was developed and became robustly used first in animals, non-human animals, then in humans. And there's some evidence of higher conditioning rates, but in part that's because spontaneous blink rates are associated with high dopamine tone. So some people actually just look at blink rates as an estimate of dopamine tone. And if in the 60s when these people were being studied and they weren't being treated, you would expect high blink rates at the baseline or spontaneously. And if you don't account for that carefully, you can misattribute spontaneous blink rates to conditioned responses that happen to be occurring during the window where you're looking. Impairments are more often seen in eye blink conditioning, and there's a number of studies there. And there have been some failures to find this. Jerry Kent, one of former graduate students here in Indiana in my lab, did a nice review paper of this that you can see. There are ways that you can look at this, and I think I can skip over that and just show you that what happens is that as time passes here, the tone comes on, and initially the tone does not generate much in terms of neuromuscular activity. The early trials are here, and early in the presentation of the stimuli, some people blink at the onset of the tone. So shortly after the tone comes on, you can get kind of a startle reflex that happens. But what we're looking for, and after the air puff comes on, I should say here, the air puff comes on, you get a very robust blink response. Sometimes people kind of move their head a little bit. Our electrodes are placed right here underneath the eye, so we're not bothered by the muscular effect in the neck. But it's this natural startle reflex to withdraw from the presentation of that air. What we're looking for is an eyelid closure that precedes the onset of the air puff. And that can be seen in this figure a little bit better. The air puff is coming on here, And what we can see is over trials, from the early trials to the second block of trials, that there's an eyelid closure that happens that precedes the onset of the ear puff. And that's what we're measuring. This is the primary dependent variable. And that can be characterized in this way. We can look at the percentage of conditioned responses that occur in that window. And we can see that individuals who are healthy controls show robust learning. And although these groups start off a little bit different, this is not statistically different, but it becomes statistically different in the last trial blocks, the later trial blocks. You can see that people with schizophrenia have impairments in this. They extinguish this response overall at a fairly similar rate and end up back where the controls and they were themselves earlier. We see this problem not only in schizophrenia, but in schizotypal personality disorder. So this is the control group again. This is a schizotypal personality disorder group, and this is a schizophrenia group. We've seen this problem in first-degree relatives, suggesting that the problem may not be associated with the manifestation of the disease, but maybe also for the risk of the disease. So this is the control group here, and then this is a group of schizophrenia first-degree relatives showing the impairment in the percentage of conditioning across the learning paradigms. These conditioned responses are associated with intellectual functioning. So if you look at the WASI IQ estimate, in the control group, there's a relationship there. And if in fact the cerebellum is playing this coordinative role, not just with motor processes, but in a variety of processes, maybe you would expect something like this. Andreasen postulates that these types of relationships may exist. We don't see this in the schizophrenia group, as if the connectivity of the role that the cerebellum is playing is disconnected or broken with regards to cerebrum and schizophrenia. So is there evidence of cerebellar impairment in schizophrenia? Yes. Using this cerebellar dependent task, yes. We've also done some work. Daejin Kim, research scientist in our lab, has looked at the modularity, and this is using FA, or fractional anisotropy. And when we look just at the cerebellum, we see in the normal controls that there's a architecture that is different here. Particularly in the vermis, you see that the module 1 in the control group is different than module 1 in the schizophrenia group. So some modularity differences, structural differences. If we look at FA, which is an fractional anisotropy, which is indication of sort of white matter integrity, these lateral regions of the cerebellum, this is CRIS2 region of the cerebellum, are associated with symptoms. So better FA, better integrity of the white matter tract is associated with reduced symptoms, reduced positive symptoms, reduced general symptoms. And this is the case whether this is reduced negative symptoms and reduced composite score. This is the case on the left and the right. So a general trend to see that better structural integrity of the cerebellum is associated with less symptoms in the disorder. If we look at what's happening during eye-blink conditioning and cerebellar activation, Jerry Kent published a pilot study with just really 2 handfuls of individuals with schizophrenia, and showed that controls have greater activation of the deep nuclei, that final output location of eye-blink conditioning, compared to schizophrenia patients. And that this activation in CRIS2, when we saw activation in schizophrenia, was associated with higher IQ scores in schizophrenia, that cognitive relationship popping up here. So when we do see activation in schizophrenia, although it tends to be impaired, that activation that we see in schizophrenia tends to be associated with better cognitive functioning. And why is CRIS2 important? I just wanted to use this to remind myself that CRIS2 is one of those regions that is directly connected with frontal regions of the cerebrum. Jerry's work in that same paper showed that this CRIS1 region, so adjacent to CRIS2, that when we see activation there, and again, I want to say that in general, there's a decrease in cerebellar activation in this task in schizophrenia, but when we do see activation, it's correlated negatively with positive symptoms, as we saw with the FA values in a different study. And same negative correlation between schizophrenia, cerebellar activation, and negative symptoms. Most recently, Nancy Lundine's dissertation work, which is now under review, with a group of 30 schizophrenia patients and 40 controls, shows less activation in cerebellum than in patients. Well, this is early paired trials and late paired trials. This is the activation in the control group only, actually, because we just don't see much activation in the schizophrenia group. But I just wanted to illustrate through Nancy's slide that it's this CRIS1 region where we're seeing quite a bit of activation and some in lobule 6. Nancy showed that when we do an ROI of lobule 6 and CRIS1, that the healthy controls have more activation than the schizophrenia patients across these various contrasts between early eye blink conditioning, late eye blink conditioning. And it's significant later in the learning trials in this particular analysis. And then Nancy, in a separate study, found correlations with some cognitive and motor functioning measures. So the activation in lobule 6 was associated with increase in letter number sequencing, and the activation in lobule 6 was associated with a decrease in PANSS positive symptom scores. And we see that both in lobule 6 and CRIS1, these regions that are associated themselves with some frontal regions as well. So that's the eye blink conditioning. I just want to, by showing these slides, say that we see similar problems with finger tapping. And so Alex Musatouks has studied the finger tapping circuitry, shown that the cerebellum is heavily involved in the temporal patterning and the following of a paced finger tapping task where you need to follow a pacing tone. And I'm going to skip over the details of the task and just show that she's demonstrated that the problem in schizophrenia is not the activation of the cerebellum per se, but the problem in schizophrenia is that there is a weakened effective connectivity in a modeling design. There's weakened effective connectivity between cerebellum and motor regions that we think are responsible for generating response. So that region that's supposed to be helping you time the response, the cerebellum, is not getting its signal to the region of the brain that's initiating the response. And this is illustrated by pathways that are decreased in schizophrenia compared to the healthy controls. We've also demonstrated using PRISM adaptation. This is another cerebellar task, that there are problems in the motor system. And in this particular task, what happens is the participant is asked to sit and look at an item that they're pointing at. And they're able to see their finger to begin with. They're asked just to put their finger at a starting point, then reach out and touch near this object. Put their finger back and then touch again at this object. And they're looking and getting a sense for the apparatus. We then put PRISM lenses on that displace their vision. And what happens is that we don't allow them to see their finger. We cover the last part of this board and they can't see their finger, but we measure where their finger is by putting a magnet sensitive electrode on the tip of the finger and measure it in a magnetic space. And we reliably displace their reaching with the goggles. With the goggles still on, we then allow them to see their fingers, and they begin to adapt. You can see an adaptation process that happens. And by the way, this is the schizophrenia patients and the control patients here. The cerebellar part that is measured is this part, post-adaptation. When we allow them to adapt by showing them where their finger is, they can get closer and closer to pointing at the right spot. But if we then block their finger again and see whether that's held, has that feedback system held? Is the corrective forward feedback system working? The answer is not as well in schizophrenia. They revert to reaching more to the left again, back over to the far left, compared to the controls who are able to keep this updated cerebellar model in place. When we look at after effects, we take the goggles off. They swing to the left, to the left, I guess it is. And when we ask them to reorient, they can go back to where they were before. But this is quite cerebellar in nature, and we have deficits here in the patients groups. So taken together, there's a lot of evidence for these cerebellar problems in schizophrenia. And let me just take 2 or 3 minutes to just illustrate a couple of psychopharmacological manipulations that we've done. One was to give a drug called secretin. And we did this in a double-blind, placebo-controlled manner to 27 medically stable schizophrenia patients. We administered this drug at baseline, and then 2 hours later, we believe this to be a fast-acting, and we believed it crossed the blood-brain barrier. It was administered subcutaneously. 2 hours later, we measured eye blink conditioning, and then 24 hours later, we measured eye blink conditioning again. So 3 measures of eye blink conditioning. What we speculated was that secretin is generated, synthesized by the Purkinje cells. It's then released and binds to a presynaptic cell that facilitates the inhibition of Purkinje cells. And if you recall, I said that inhibiting the Purkinje cells allows this release of their inhibition on the deep nuclei so that you can generate blinks. So we were trying to affect this circuit here and allow the interposed nuclei to generate the response that should be generated by removing the inhibition Purkinje cells have on this. And what we saw was at baseline, the baseline matched groups, we selected groups that were matched on baseline performance, that the secretin group showed an increase in eye blink conditioning with no change in the placebo group. This work has been corroborated subsequently by direct infusion into the cerebellum in rats. And it turns out that, they see something very similar as we saw in the human study when they inject this directly in the cerebellum in rats. We did this again with N-acetylcysteine, and we postulated an increase in extracellular glutamate, increasing the LTD and the LTP at the deep nuclei. And what we saw was something similar. At baseline, we selected a group that were matched at baseline, and the N-acetylcysteine group was going up. Same sort of mechanism generally is in play here, we think. So there's some evidence of some psychopharmacological probes that are going directly after this circuit having positive effects. And again, we think acting on these LTD and LTP mechanisms that are well established in the animal models. So in summary, I've shown you that simple Pavlovian conditioning procedure that measures a fundamental associative form of learning is impaired in the schizophrenia spectrum. First-degree relatives, people with schizotypy, and individuals who've developed the disorder. And this is the evidence in schizotypal personality disorder and first-degree relatives. Healthy controls, but not schizophrenia patients, seem to have a relationship between these associative learning measures and cognition. So this absence of that relationship in schizophrenia may indicate failures of these circuits and failures of the structural integrity of these regions. The MRI analyses revealed impairments in white matter microarchitecture or microstructure, and that these were associated with more severe symptoms. The cerebellar bold activation was impaired during conditioning in schizophrenia. But when there is cerebellar activation, we see that this is associated with increased cognitive ability and decreased symptoms of the disorder. And then we showed in a couple of other tasks, finger tapping and prism adaptation, that we see similar cerebellar dependent abnormalities. So we think that the neurochemical studies that we're beginning to do, and we're trying to do some micro stimulation using TDCS, for example, we think that the circuit may be suitable for some interventions, as is being done by other groups, particularly De Leon at Harvard is doing some micro stimulation, brain stimulation. So that's the story, and we can maybe take just a couple questions with remaining time. Thank you all for your attention. And again, thank you very much for the invitation. Very happy to have the opportunity to do this. It's an honor to speak with you.
Thank you, Dr. Hetrick, for a wonderful presentation here today. We've had a few questions come in already. I'll keep an eye on if more do. First question that came in is, how might the cerebellum play a role in the autonomic and immunological functioning changes associated with psychotic disorders?
Wow. I've not thought about that. I'd be curious, really curious to know what motivates the question. I imagine there may be some knowledge there that's motivating that question. I know that the cerebellum plays a role in very low-level coordinative processes, interoceptive processes, balance and coordination through low-level regions, right? I don't really know a lot about autonomic control of cerebellar nuclei or cerebellar processes. Is there a way to find out what motivated the question? I'd be curious.
I'll see if we get more info in the chat here, and we'll maybe circle back as it comes in.
Yeah.
Next question. You commented on possible pharmacological interventions targeting cerebellar mechanisms. Do you see a place for psychosocial interventions for schizophrenia that may also target these mechanisms?
That's a very good question. And I don't know if Jaime Morales is on, but graduate student in my lab, Jaime Morales, did his qualifying exams looking at basically social neuroscience and social processes and the extent to which the cerebellum may be playing a role in that. There have been some evidence from animal lesion studies that lesioning areas of the cerebellum leads to emotional ability, for example. And so thinking about social modeling, the modeling and the fluidity of social exchanges, Jaime is beginning to think about, and there's a developing literature actually on how social processes may be impaired when cerebellar impairments exist. And what that means, I think, is I'm a strong believer that behavior changes the brain. We don't need drugs to change the brain, although they do. But behavior changes the brain. We can modify circuits by highly practiced behaviors. And so some people now are doing basically motor and some non-motor interventions to modify cerebellum, to strengthen cerebellum. And showing that there are some benefits to practiced cerebellar tasks on what we don't typically think of as cerebellar functions, these more cognitive processes. So I think there is some hope there, but probably the next generation of cerebellar researchers like Jaime can take that stuff on.
Sorry, I was on mute there. Jaime offered to say a little bit, if you'd like him to join on here.
Yeah, that'd be great. That'd be great.
Let me see, Jaime, if I can unmute you here real quickly.
Jaime is just upstairs, I see. Here in the building.
Well, hopefully we won't need him to shout down.
Right. What did you want to say, honey? There we go.
Okay.
Everybody.
Jaime, you should be able to unmute.
Yep, here. So as Bill said, there's some initial findings with specifically more in cerebellar ataxia, so people that have lesions in the cerebellum, showing that they do have focused areas that lead to emotion recognition deficits or theory of mind deficits. So some of the literature points, or at least theory of models point towards an interaction of how the cerebellum predicts what our actions are going to be in the future. Through this kind of modeling, we can begin to understand why the cerebellum might come into play when it comes to social interactions. Given within social interactions, we need fluidity and efficiency to occur for us to understand what one person is saying or what one person is expressing, and dovetail that with our understanding of how interaction should go. So my line of research is trying to understand if the cerebellum does play a role in psychosis, given, as we know, the surmount of social cognitive deficits in psychosis populations, to see if a lot of the efficiency or inefficiency that's occurring within these populations is due to some predictive modeling deficits that could be led by the cerebellum, but that could be at least influenced by cerebellar processes. And there are specific areas within the cerebellum that have been, I'm sure Bill talked about, that have been associated with non-motor cognition, such as CRUISE-1 and CRUISE-2, which are thought to be the more non-motor cognitive areas where either efficiency models or prediction models are elicited. I can go way more into this because I did my quals, but I don't want to take away from Bill's questioning time. So thank you for having me.
Thank you, Jaime.
Yes, thank you, Jaime, for being willing to jump in for the discussion here today. I appreciate that.
Same here.
Unfortunately, we are at the end of our time here. So I'd like to thank you again, Dr. Hetrick, for a wonderful presentation. And thanks everyone for joining us here today. Hope you'll be able to join us next week, where we have Sarah Kapelovich from University of Washington talking about cognitive behavioral strategies for use in family psychoeducation among caregivers of individuals with first episode psychosis. So we hope everyone stays well and stays healthy, and we look forward to seeing many of you next week here back in our presentation. Take care, everyone.
Thank you. Bye-bye. Have a good weekend.